In biopharmaceuticals, speeding new molecules to market is crucial, and achieving target purity and yield often requires more than the standard 2 to 3 step mAb purification process, necessitating modifications or significant downstream process development. I'm Miles Skoda. This is the bioprocessonline.com webinar series, and today we're talking building a purification toolkit for an expanding variety of mAb therapeutics. Now, joining me for this presentation are three industry experts from Thermo Fisher Scientific. First, Chantel Gaskin, Field Application Scientist for Purification, Carlos Ferrero, Field Application Scientist for Process Liquids, and Ryan Hubble, Product Manager for Process Liquids, who together are going to look at strategies for building an effective downstream processing toolkit, discuss matching purification of various mAb formats to a wide array of chromatography resins and buffers, and look at enabling optimal integration of these resins and buffers into mAb purification processes. So we will be holding a Q&A session following this presentation. Please feel free to submit questions as we go along. We'll get to as many as possible during that Q&A, and any that we don't get to, I'm going to pass along to the team for potential follow-up after the event. And then just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often much less. You'll get an email with a link to it, or you can just use the same link that you used today. So with that said, I'm going to hand it over to Carlos to get us started. Carlos?
Thank you. Starting with the capabilities of our bioproduction group, we support developers and manufacturers of biological-based therapeutics and vaccines with a portfolio of premium solutions and services focused on upstream cell culture, downstream purification, analytics for detection and quantitation on process and product impurities, and a suite of single-use solutions spanning the biologics workflow. The focus in this webinar is going to be downstream purification, where Thermo offers a broad portfolio of high-performing resins. From the Affinity side, the Capture Select offering addresses purification of a wide range of biologics. The manufacturing capabilities for the Affinity ligands consists of vessels ranging from 10 liters to 15,000 liters, and is located in the Netherlands. For polishing, the porous ion exchange and hydrophobic interaction resins provide superior resolution and capacity for effective impurity removal. The manufacturing capacity consists of vessels up to 250 liters, located in Bedford, Massachusetts. Lastly, robust custom ligands and resin capabilities offering commercial tailored purification solutions are also available. For your process liquid needs, we offer a comprehensive buffer preparation portfolio that includes made-to-stock catalog buffers, our made-to-order buffer library, and fully customized solutions designed to streamline and support your specific buffer requirements. All our products are suitable for late-stage GMP and commercial manufacturing processes. Now, let's take a look at where the monoclonal antibody market is heading and why the tools and capabilities we just discussed are so critical. But first, let me draw your attention to the poll question showing on the screen.
Thank you, Carlos. So speaking of GMP processes, in the last 40 or so years of mAb clinical history, there has been steady growth in development of these novel therapies based on antibodies. With the movement towards personalized medicine, more focus on rare diseases, the aging population, the mAb industry really isn't slowing down on growth anytime soon. It's estimated actually that in the next 10 years or in less than 10 years, the ADC industry could double. The bispecific mAbs industry could increase 7 times. So there really is a lot of growth in antibody therapeutics. So taking a look at the kinds of molecules we're talking about, apart from your standard mAb, there are IgGs obviously with an altered FC region, FC fusion proteins, bispecifics, Fabs, and ADCs. We know that working in this type of diversity of modalities, there could be issues with purification. So, for example, species that don't bind your standard protein A resin. Also, there could be issues with removal of the different types of product and process related impurities like aggregates and high molecular weight species. So, to address this, Thermo Fisher has developed a range of affinity and polished resins. And like Carlos mentioned, the Capture Select brand is our family of affinity ligands and resins that can be used to purify a wide range of these types of molecules. Then we also have our high capacity porous resins that are used for intermediate and final polish chromatography. And that includes various chemistries that I'll go into a little bit later in this talk. But first, our resins form the basis for your antibody purification toolkit. But purification process really could not work without buffers. So I'll hand it back over to Carlos to tell us a little bit more on that.
Thank you, Chantal. If we take a look at the downstream workflow on the slide, we can see how processed liquids seamlessly integrate with chromatography resins by providing ready-to-use solutions for each purification step, including equilibration, elution, cleaning in place, and storage. These pre-made buffers ensure consistent composition and control pH and conductivity profiles across batches. This not only supports resin performance and reproducibility, but also eliminates the need for time-consuming in-house buffer preparation and reduces the risk of human error. Now, I would like to draw your attention to the following poll question. Have you encountered challenges when transitioning from non-GMP to GMP raw materials during the move from process development to commercial manufacturing? As we navigate the drug development pipeline from early research to commercial manufacturing, buffers play a critical role at every stage. In early research and preclinical studies, materials, including buffers, are often on a small scale at not under GMP. However, as we enter into clinical trials, the regulatory expectations sharply increase. At phase 1 and beyond, GMP-grade buffers become essential to support reproducibility, compliance, and safety. By phase 3 and commercial production, the need escalates to large-scale GMP-compliant buffers that support both process control and regulatory approval. This is where our pre-made GMP buffer offering aligns perfectly. Whether you are in drug development at moderate scale or manufacturing at commercial volumes, we help ensure that key raw material decisions like buffer quality and consistency are addressed early and effectively to de-risk your journey to the market. One of the most strategic decisions you can make in drug development is the early adoption of GMP-grade materials, especially buffers. By using GMP-compliant buffers from the beginning, you align your process with regulatory expectations, significantly reducing the risk of delays down the line. You also simplify the transfer and scale-up as your materials are already aligned with commercial production standards, reducing rework and revalidation. From a cost and time perspective, this minimizes the need for compatibility studies between non-GMP and GMP materials later on. And as the image on the right shows, even a slight change in raw material quality, like buffers, can have an impact in critical quality attributes and ultimately your product's performance. Early investment in GMP buffers helps secure a faster, smoother, and more compliant path to market.
Thank you, Carlos. So that's right. It's very important when considering building quality into your process to think of these things, right? So when starting out in your process development, There's several types of purification tools that you can consider, and we'll go into that next. So as I mentioned before, we have a variety of affinity resins for purifying all the different types of antibody-based molecules, Fabs, bispecifics, as you can see here, light chain binders, binders, FC binders as well. All of this is apart from your traditional protein A standard resin. Our Capture Select resins are based on camelid VHH fragments that are screened initially for their specificity to a target molecule, the stability of the ligand, as well as mild dilution profiles. They're produced recombinantly in yeast, making our Capture Select ligands a really good option for using in a scalable process development. And this is really for all stages of development and manufacturing. Starting with our CH1XL resin that binds to the CH1 domain. Many of our Fab customers use this for removing light chain, free light chain species, as well as host cell proteins. It has a DBC of about 19 grams per liter and provides efficient elution at a mild pH, around pH 6. So you can use CH1XL, either an agarose version, but we also have the magnetic bead version for those looking for sort of early research phase purification, or for those customers that have automated platforms like the Kingfisher that can handle magnetic beads. Now for our light chain binders, we have our Kappa and Lambda XP, which were designed to bind 100% of Kappa or Lambda subtypes. Both have high dynamic binding capacity for IgGs and will bind around 30 grams per liter of Fabs. And our FCXP resin binds the FC region away from the protein A binding site. So this is a good option for not only mAbs where the protein A site is altered, but also FC fusion proteins and bispecifics. It's also a good choice for IgG purification from complex sources like biological fluids, plasma, and such. It has a high dynamic binding capacity, and it's available in both agarose and polystyrene DVB backbone. So there are options that different options you can evaluate depending on the nature of your feed material, as well as the plans that you have for scale up. And lastly, our newest protein A resin, mAb Capture C. This ligand has retained the protein A binding sites, traditional protein A binding sites on the IgG. So specifically the one at the CH2-CH3 interface, but also it can bind some VH3 fragments. mAb Capture C also has high binding capacity, but the nice thing is that it's also alkaline stable that allows for rigorous cleaning. So we performed a life cycle study based on yield and found stable recovery of around 90% for 2 different molecules for over 100 cycles. This included 2 15-minute sodium hydroxide cycles each. And for simplicity, as you can see on the left-hand side at our study, the run outline, the run conditions, we used common buffers like citric acid and PBS that are also available as pre-made buffers, what Carlos mentioned previously. So we've reviewed ways you can begin building a purification toolkit. Depending on your type of mAb or antibody-based molecule, there are several affinity resins to select from for developing your capture step. And whether you need a light chain binder, like I mentioned, Kappa, Lambda, XP, or you need maybe something more selective of intact Fabs like CH1XL, or even if your molecule does not bind traditional protein A resins, you can opt for the FCXP resin. So our Capture Select portfolio allows for high purity in a single step. And this obviously reduces the need for extra process steps and increases productivity. And also to help further, I did want to mention that your regional field application scientist is available for providing recommended process conditions to get you started. Now, we also offer a range of polished resins to include in your mAb purification toolkit. And as we touched on earlier, buffers play a huge role here as well in establishing optimal conditions for your polish steps. So I'm going to hand it over to Carlos again.
Thank you. As we progress through purification, one of the biggest challenges is removing impurities while preserving product quality and yield. Buffers play a central role in addressing this challenge, whether it's hostile proteins, DNA, aggregates, viruses, or low and high molecular weight species. The right buffer conditions with pH, conductivity, and additives are what enable effective separation and impurity clearance. This is why buffers aren't just support materials, but they are critical tools in purification process development. By leveraging buffer-mediated solutions tailored to its impurity class, we help ensure consistent high-purity output that meets regulatory expectations and performance standards. Now that we have seen how buffers are essential for impurity removal, regulating parameters like pH and conductivity to selectively target contaminants, the next logical question is, how can we design these buffers effectively and consistently? That's where this buffer calculator that you are showing on the screen becomes incredibly valuable. By simply inputting the buffer species, salt, and target pH, this tool calculates the exact quantities of each raw material required. This ensures precise and reproducible buffer preparation. This is critical not only for consistent purification performance, but also during buffer screening, when evaluating multiple conditions to optimize your process. Using this tool, you can rapidly generate standardized buffer recipes, accelerating process development and minimizing variability from the start. Now, I will hand it over to my colleague Chantal, who will walk you through additional process calculators that can support your purification toolkit.
That's right. Yes. So we also have these other calculators to help with streamlining and simplifying the calculations that you need to carry out during your process, your purification process development. So using the same link that was on the previous slide, you can access our column dimensions and column efficiency calculators to do your column packing calculations. And then our process modeling calculators really cool. It ties everything together. It gives you suggestions for resins, buffers, and process conditions, all based on the desired molecule type that you input into the calculator. Now, switching back to polish chromatography. So our Poros brand of resins are made with polystyrene divinylbenzene, polystyrene DVB. And this provides a chemically resistant and rigid backbone for our polished resins. They come in a convenient 50 micron diameter bead and have a range of chemistries, including anion exchange and cation exchangers that you see here on the screen, which you can use depending on the different properties of your molecule and your targeted molecule, as well as any impurity molecules that you're trying to remove. Our Poros HQ, which has a mixed amine chemistry and a unique binding profile, is widely used in flow-through mode for many mAb processes. It's used for viral clearance, removal of HCPs, host cell DNA. Our Poros HS is our strong cation exchanger, also used in many mAb processes for removal of high molecular weight species in bind dilute mode or overload mode. And our Poros XQ and XS are high salt tolerant versions of our strong AEX and CEX. And these resins provide higher binding capacity and a wider range of process conditions. We also have 3 HIC resins covering the extreme ends of hydrophobicity, with our porous ethyl on the lower end, often used in bind dilute mode, and then our porous benzyl and benzyl ultra resins used mostly for aggregate removal in flow through mode. Also can be used in bind dilute. And lastly, our porous caprolate, which is our mixed mode resin based on caprylic acid chemistry. This resin was designed for feed streams with high aggregate levels, so thinking 10% aggregate levels or more. Porous caprolate combines CEX and HIC chemistry for maximizing capacity of high molecular weight species in flow-through mode specifically. And since it is on the porous backbone, it has a linear pressure flow curve and large through pores, so it's really useful for increasing productivity in your scalable process development. Now, we performed a series of experiments to test the ability to remove aggregates and other impurities. So next, I'll review some data that we generated in our DOE and load density studies, as well as I'll also summarize some impurity data that were generated. For the DOE study, we looked at monomer recovery and aggregate in the flow-through as a function of pH and conductivity. The sample here that we used contained 10% aggregate, and it was loaded at 100 grams per liter onto a 1 ml column. The contour plots that you see here show that monomer recovery increases with pH and conductivity. But interestingly, on the right-hand side, there's a sweet spot with respect to conductivity and pH that would result in less than 2% aggregate in your recovered material. So that's the areas in light green and blue here on the right-hand side between pH 5 and 5.5. And we see that regardless of salt concentration. So focusing just on the one point within that window of operating conditions that we saw previously, we performed a low density study to assess the capacity of aggregate just at that one point. We selected pH 5.25 and 275 millimolar sodium chloride. Again, using a 10% aggregate feed and a total of 325 grams per liter load, we found good recovery at 85 grams per liter that you see here on the right-hand side with just a 1% aggregate. But if you could accommodate just 3% aggregate, you can max out on your monomer recovery. So we saw the highest monomer recovery is achieved at higher loadings. We also did confirmation runs at 3 different pH and conductivity conditions at greater than 150 grams per liter loadings. So the representative chromatogram that you see here on the right shows the load and chase steps, followed by a cleaning step with 0.5 molar sodium hydroxide. The SIP peak that you see here contains the aggregates bound during the load. Confirmation runs, all 3 confirmation runs showed consistent reduction of aggregate from 10% to in the load, 10% in the load to about 2% based on SEC HPLC data. There was also consistent monomer recovery of at least 90%. Lastly, here's a summary of the impurity clearance. So greater than 95% host cell protein clearance, greater than 90% leach ligand clearance across all the conditions that we tested. And I did want to mention that we have more in-depth analysis of the different host cell proteins that were cleared. So if you are interested in that, reach out to your local FAS and they have more information for you. Now, speaking of DOEs and different buffer conditions, we have a range of pre-packed tools that could help your screening experiments. Most of our Capture Select and porous resins are available in pre-packed 1 ml and 5 ml columns. And we also have the 200 and 600 microliter robo columns available that could be used with your liquid handler, so your TCAN or your Hamilton. We also have a selection of Affinity resins available in 96 well filter plates that can be used with a vacuum manifold or even a centrifuge plate, a centrifuge that has the plate rotor on it. We provide protocols and guidelines for each of these formats to help you go from small scale screening experiments to lab scale and beyond. So I did want to draw your attention to the next poll question here. Based on your lab setup, what is your preferred tool when getting started on purification process development and screening? Now, these tools are focused on screening specific resins and buffer conditions. To make things even easier, I'll pass it on to Ryan to highlight the convenience of ready-to-use buffers and how they can aid in your mAb purification toolkit.
Thank you, Chantel. So outsourcing your process liquids can offer several benefits to streamline your operations. Shifting from in-house production of buffers to outsourcing can simplify changeovers, reduce the total handling of raw materials, increase manufacturing agility, free up floor space in your facility, most importantly, put your people and capital purchases to better use. You can have your technical team focusing on producing your core products rather than just generating components. One significant challenge that companies face in preparing their own buffer formulations is the management of raw materials. This becomes a big problem for complex formulations as the number of suppliers of raw materials increase. The overhead associated with ordering, warehousing, stocking, and producing materials can be substantial. Resources are stretched thin, and the ability to be agile decreases. Purchasing ready-to-use buffers can remove these hurdles and allow you to focus on your primary objectives. And we offer a range of solutions, from stock catalog buffers to fully customized formulations, all stored in single-use flexible bioprocess containers. Our packaging options range from 1 liter to 1,000 liter and can be customized to meet your needs, including tubing, connectors, and the film. So the last tool we can provide is storage and inventory management. We all know that production schedules can fluctuate and that warehouse storage can be limited. We can help with these challenges by storing buffers off-site and delivering them when needed. This would reduce the in-house storage costs and constraints. This option could also reduce potential expedited shipping fees. Thermo Fisher offers up to 90-day ship date deferment for regular orders and a stocking program for buffers with consistent quarterly forecasts. And the implementation steps are simple. Once we confirm the quarterly forecast, we set reorder points, sign the stocking agreements, and maintain stock levels through ongoing reviews. So there you have our complete purification toolkit for mAb-based therapeutic development. Today we covered the Capture Select Affinity matrices for mAb purification, the porous polish resins for impurity removal, pre-made buffers and concentrates, convenient chromatography resin formats, and ongoing field applications support for all your needs. These solutions not only enhance efficiency and consistency, but can enable you to focus on your core competencies, driving forward the development of therapeutic solutions. Thank you for your attention, and before we get into Q&A, we have one more polling question for you.
All right, awesome. Yeah, thanks, Ryan. And yeah, this poll question is up on your screen, everybody. Thermo Fisher's here to help you. Would you like to speak to a technical specialist about building your purification toolkit? Let them know. Response right there on the poll, and we'd appreciate letting us know. And so with that, we're going to get into the Q&A. Audience members, you can submit questions again through that Q&A box on your screen. We're going to get to as many possible as possible here. And then any we don't get to, I'll pass along to the team for some potential follow-up as well after the event. And then in addition, take a look at your screen and make sure to answer any of the poll questions that are still up there, any of the survey questions, and then, let us know for sure on those, and then any resources available as well. If you wanted to reach out to our speakers directly, you can contact them. Should be on the right side of your screen. There's a speaker bio panel. You can click on the little blue circle inside below each of their names, and that'll let you reach out to that speaker directly in case you have any more specific questions that you might not want to ask here. And so with that, let's get into these questions. I'm going to go to this first one. This one is coming in from Tara. So asking, a lot was said about buffers for purification steps. But in reality, what buffers do you offer for steps like equilibration, elution, strip, or cleaning for mAbs?
Well, thank you for the question. We have a different array of buffers that can be used depending on the chromatography step that we're talking. For equilibration, we can support you with the basic 20 molar 3s or PBS at a physiological pH between 7 and 7.5. In elution, we can support you with 50 millimolar sodium acetate at a more basic pH. Citric acid as well at 20 molar. Also for strip buffers, we can support you with whatever formulations you need. For instance, 0.1 molar phosphoric acid, 0.1 molar glycine. For cleaning, the standard ones will be sodium hydroxide, 8 molar urea, citric acid, 0.5 molar. That would be more the standard formulation for each step, but we pretty much can support you by our catalog, buffer library, and as custom buffers. For storage, most likely you might be needing 20% volume per volume ethanol. We can support you through the catalog and also the buffer library options there. So supporting different packaging options. So we have worked to really integrate all these PLPs, really to use buffers into chromatography resins. So the possibilities are infinite there. Thank you.
Awesome. That's great, Carlos. Thanks so much. Yeah, a lot of stuff there. Let's see. We've got time for a few more. Let's get on to this one from Mikhail. So could you elaborate more on how to use the 96 well plates for mAbs or biospecific mAbs process development?
Yes, good question. So typically we use 96 well plates in one of two ways, right? So either you're going to be, in the case of when you have like a pipeline of many different constructs, whether they're mAb or bispecifics, whatever the molecule is, you have a lot of constructs. You might screen the ability for the resin to bind those constructs. So it's really a screening of the resin to make sure that you have binding of your molecule, or you can the other way to use them is to basically test multiple buffer conditions, like I mentioned in the case of caprolate. So when you have multiple buffer conditions, like wash buffers or elution buffers, right? And the nice thing is that we've included plate layout suggestions, suggested protocols, and that type of thing, guidances in the product manual. So you can always refer to those, but you could always work with your local FAS as well. They're always more than happy to help you design a more custom study for your project. Thanks for the question.
All right. Yeah, perfect. Thanks, Chantal. That's great stuff. Let's see. Another one here. So this one's coming in from Jun. You highlighted the case study of using caprolate mixed mode resin in the removal of aggregates and HCPs. He's asking, sometimes we have difficulty with removing specific HCPs like lipases and clusterin. Do you have any suggestions for what tools to evaluate for that purpose?
Yes. Well, thank you again for that question. Actually, yes, I did mention that so our colleagues in our R&D department have performed a more in-depth analysis of the different HCPs that were cleared with the porous caprolate. I believe lipase and clusterin were among those HCPs that were identified in that study, but there were hundreds of HCPs identified. And the study was published, actually, so you can reach out again to your local FAS to ask for the details on that publication. But I would recommend either using mAb Capture C, or if there is an issue with binding onto protein A, you can use our FCXP resin for capture, and then followed by the normal low pH hold for viral inactivation, and then load that elution pool onto porous caprolate. So depending on the elution conditions from your capture column, you may need to adjust the pH prior to load, but that's what I would suggest. Hope that helps.
All right. Yeah. Thanks again, Chantal. That was great. All right. Another one here. This one coming in from Hassan, kind of a more general one. So how can outsourcing processed liquids make processes more efficient?
Yeah, I can
Carlos,
take
you're on
this one.
mute. Oh, gotcha. All right. Perfect.
I can take this one, Carlos, unless
Sorry.
you want it, since you took the last
Go
one.
for it. You go for it.
Yeah, I can take this one. So basically, outsourcing your process liquids can free up critical space. So this eliminates the need for in-house prep areas, so tanks and skids. So you can have smaller, more focused facility layouts. Additionally, reduces operational bottlenecks. So again, removes the buffer or media prep from the critical path, which is often a constraint when you have an intensified workflow. Improve consistency and quality. So outsourced solutions they're all made under GMP conditions with tight specs. So that reduces the variability from batch to batch that you might see internal manufacturing. And ultimately, it enables leaner, more agile operations. So supports single-use, modular, and flexible manufacturing setups, which is key for intensified or if you have multiple product lines. And ultimately, it can simplify your scale-up and tech transfer. So again, standardized vendor-supplied liquids. This makes it easier to replicate processes across sites or with CDMOs.
All right. Perfect. Yeah. Thanks, Ryan. Let's see. So we've got maybe time for one more here, I think. This question's from David asking, how do you use these purification tools to ensure the scalability of your purification processes from small bench scale experiments to large scale production batches?
Yeah, well, one way to do this is I like to keep the end in mind, right? So it's helpful to know what your final scale will be so that you can design your scale down model appropriately. So the basics, remember to maintain load and elution residence times. One easy or convenient way to do this is to use our pre-packed GoPure columns, right? They're available I mentioned the 1 and 5 mil columns. Those are at 5 and 10 centimeter bed heights, right? So those are usually pretty convenient for scale down models. Other than that, all the resins I spoke about today are manufacturing grade. So they come with documentation to support regulatory submissions. So you really don't have to worry about the resin quality or trying to switch, to resins with appropriate quality documents when it's time to scale up. So those are good to go.
All right, perfect. Chantal, thanks so much. And I think this is probably going to be a great place to wrap up. And so kind of with that, again, Ryan, Carlos, Chantal, thanks for joining us today. This was an awesome presentation, a lot of great insights, and thank you for getting to these great questions from our audience. We do have a few more questions left here that are kind of specific, and then a bunch of other ones that I'm going to pass along again for some potential follow-up from the team here. And so if we didn't get to your question, make sure to keep an eye out. But otherwise, we hope that everybody does have a great rest of your day. Thanks again for joining us, audience members, and we'll see you next time. Bye, everybody.